which deals with the skeletal system. So bones are the organs. Again, they're made up of more than one tissue. The skeletal system is composed of many different tissues, including bone tissue, cartilage, dense connective tissue, blood, and nervous tissue. It's important to remember that bones are alive. We have a tendency to think of bones as the samples we have in the laboratory or the kind of things you see on TV. Those are bones from individuals who are no longer with us. A bone in an organism is alive, definitely, and is very multifunctional. There's a lot of things that go on a bone besides just providing levers for us to move around. They support and protect softer tissues. In fact, some of our bones are going to be designed just for that, not for movement at all. They provide points of attachment for muscles. Those do act as levers for our manipulation of the environment, movement through the environment. One thing you may not be aware of is that our blood producing cells are all found in bone. All of your blood originates in bone tissue. And they are also very important for storing inorganic salts. Bones of the skeletal system are different from one another. There are approximately 206 named bones in an individual, but they're all different in sizes and shapes. All bones do have similar structures, however. They develop in one of two ways, and their functions are very similar despite their size and shape. So we can classify bones based on their shape, in which case we have long bones, which are long and narrow, longer than they are wide. They tend to have expanded ends. Many of the bones, the arm bone, leg bones you would think of, are long bones. But it is important to remember that the length of the bone doesn't define it as a long bone. There are some very short, long bones, especially in your fingers and toes. Short bones, on the other hand, are more cube-like. Their length is roughly equal to their width. These would include the sesamoid bones, which are found within tendons, tend to have a rounded shape to them, and flat bones, which are plate-like and have broad surfaces. And if you can't fit it into long bone, short bone, or flat bone, we have the catch-all category of irregular bones. They have a variety of shapes. Most of them are connected to other bones. So in a long bone, there are some parts, and I will go over them now. You'll learn more about them in the lab on Thursday. There is the epiphysis, which is the ends of the bone. There's a proximal and a distal epiphyses. There's a diaphysis, which is the shaft. The metaphysis is between the diaphysis and the epiphysis. It's a widening part. Articulate cartilage is going to cover the epiphyses. We don't see that in our preserved bones. There's also periosteum that encloses the bone. It's a dense connective tissue layer that again is gone from the bones that we have in the laboratory. Bone itself can be either of the compact type or the spongy type. The compact bone is going to be found in the wall of the diaphysis. It's also called a cortical bone. It has the typical bone structure, the lamella structure that we'll look at a little later on. Whereas in the epiphyses we have what's known as spongy bone, which has trabeculae in it instead of lamella that we see in the compact bone. Within that diaphysis there is a cavity, a hollow chamber, where bone marrow is found. And in young people, that bone marrow is red bone marrow, actively producing red blood cells. But as we get older, in the long bones, that tends to become yellow marrow and is used more for storing fat. The endosteum is going to line that space of that medullary cavity, as well as other cavities, spaces within the bone. And again, bone marrow can be red if it's actively producing blood cells, or yellow if it's storing fat, and that would be found within that medullary cavity as well as in some of the spongy bone spaces. Microscopically, a mature bone cell is referred to as an osteocyte. It occupies a chamber within the bone called the lacunae. Osteocytes exchange nutrients and waste through cell processes with teeny passages, and those are called caniculi. The extracellular matrix of bone is mostly collagen fibers, as well as inorganic salts, and those inorganic salts can be used by the body when those are needed. Collagen is going to give bone resilience. Resilience we don't really have with these prepared bones. Or they're not prepared, these preserved bones. And inorganic salts make the bones hard. So when you look at a bone in the lab, you're seeing mostly the effects of inorganic salts. The collagen has dried up within those bones. Compact bones has cylindrical units called osteons. Those are strong and solid. They're used for weight bearing. They're very good at resisting compression. Whereas spongy bone has branches of plates called trabeculi, and give it a little bit of flexibility, and the space between them reduces the bone's weight. And all bones are going to have some combination of compact bone and spongy bone. So those osteons built from the osteocytes living in the lacuna. They come out in layers of matrix around a central canal, and those layers are called lamellae. They have perforating canals that will join adjacent central canals and blood vessels providing nutrients to the bone tissue. Osteocytes can pass the nutrients through those caniculi, and those perforating canals used to be called Volkmann's canals. Parts of the skeletal system begin to develop during the first few weeks of prenatal development. Starts out in the form of a hyaline cartilage model or a membrane model, depending on the type of bone you are forming. And that would be an intramembranous bones versus endochondral bones. Intramembranous ossification are bones that are flat, like the skull bones, the clavicle, the sternum, some of our facial bones, and they form between sheets of connective tissue, between a membrane. Endochondral ossification are the long bones and the vast majority of the skeleton, and they are formed by building bone around a hyaline cartilage model of the bone you want. So those intramembranous bones originate within sheet-like layers of connective tissues. They are broad, flat bones. Again, some examples of the flat bones of the skull, the clavicle, the sternum, some facial bones such as the mandible, the maxilla, the zygomatic. Intramembranous ossification is a process of replacing that embryonic connective tissue to form intramembranous bones. Mesenchymal cells will differentiate into the osteoblasts. Osteoblasts will eventually mature into osteocytes. But those osteoblasts are actively forming bone, depositing that matrix around themselves. When they are completely surrounded by the matrix, then they are then referred to as osteocytes, and their space they live in is called the lacunae again. And mesenchyme is on the outside and will form the periosteum for flat bones. And that's mapped out here. Endochondral bones, on the other hand, are built from a hyaline cartilage model. So they begin as hyaline cartilage. They have a general shape of the bone they are going to become. They do make up, this is how most bones in the skeleton, and for example, the femur in your leg, the humerus in your arm, radius, the tibia, phalanges, and vertebrae. This process begins by replacing the hyaline cartilage to form an endochondral bone. So it starts out as a hyaline cartilage model. Chondrocytes, those are cartilage cells, enlarge, and the lacunae get bigger. The matrix begins to break down, and the chondrocytes die. Osteoblasts will then come into the area and begin depositing bone matrix, and that osteoblast will form spongy first and then compact bone. Once they're encased by that matrix, the osteoblasts again are referred to as osteocytes. So here we have the hyaline cartilage model, a primary ossification center, secondary ossification centers. Epiphyseal plates will be important for development. That's how bones are going to grow longer as we grow taller. And we have osteoblasts and osteocytes pointed out. So in intermembranous ossification, we have sheets of connective tissue, whereas endochondrial, we have a hyaline cartilage model. Mesenchymal cells will differentiate into osteoblasts and intramembranous. Cartilage tissue will break down in endochondrial. You get a dense network of blood vessels supplying or developing spongy bone in intermembranous, whereas the blood vessels and differentiating osteoblasts in the periosteum invade the disintegrating tissue in the endochondrial. Osteoblasts will then become osteocytes when the matrix surrounds them in intramembranous. In endochondrial, a very similar thing will happen, but the osteoblasts take a little longer. They invade the cartilage. Mesenchyme becomes periosteum on intermembranous, and osteoblasts beneath the periosteum will begin to deposit compact bone in endochondrial, and osteoblasts on the inside of the periosteum in intermembranous will deposit compact bone over the spongy bone, and osteoblasts will become osteocytes in endochondrial. So I said that epiphyseal plate was important for bones to grow longer as we grow taller. And in a long bone, the diaphysis and the epiphysis are separated by a region of cartilage called the epiphyseal plate. It grows in 4 different layers. There's a resting cartilage layer, which is closest to the epiphysis. Those are resting cells that are anchoring the epiphyseal plate to the epiphysis. There is a zone of proliferating cartilage, which are rows of young cells undergoing mitosis. Then there's a zone of hypertrophic cartilage, which are rows of older cells that were left behind when new cells begin to appear. This is going to thicken the epiphyseal plate and lengthen the bone overall. Eventually that matrix will calcify and the cartilage cells, the chondrocytes, will die. Then there is a zone of calcified cartilage, which the dead cartilage cells in the calcified matrix, it's becoming more of a bone. And you can see those layers here. Osteoclasts break down calcified matrix. Osteoblasts invade the cartilage with bone tissue, and then bone can grow in length as long as the cartilage cells remain active. And when the ossification centers meet, the epiphyseal plate ossifies, and then that particular long bone cannot grow in length. We also grow in width and breadth by thickening the compact bone around the outside under the periosteum, and at the same time that occurs in the diaphysis, the medullary cavity will widen. So I will stop here for a second and bring up some terms. Osteoclasts are cells that break down bone matrix. We're going to see osteoclasts involved in homeostasis of bone. Osteoblasts we've seen before, those are the more immature cells, whereas osteocytes are the more mature cells. And those clast, blast, and cytes are going to be common suffixes for cells as we go through the year. So I'll let you work through the timetable of ossification. Homeostasis, bone modeling, is going to occur throughout our lives. There are opposing processes, deposition and reabsorption that occur in the surfaces of the endosteum and the periosteum. So bone reabsorption is removal of bone, and that is under the guidance of osteoclasts that are actively digesting that material. Whereas bone deposition is the formation of bone, and that's under the work or the result of the work of osteoblasts. And we do replace between 10 and 20% of our skeleton each year. There are a number of factors that can affect bone development, growth, and repair. Nutrition, sunlight exposure, hormone levels, physical exercise, those are all important. Vitamin D, you need calcium to be absorbed. Calcium can't be absorbed without vitamin D. Without vitamin D, we can get a couple of diseases called rickets or osteomalacia. Vitamin A is very important. It helps with osteoblast and osteoclast activity, and deficiencies in vitamin A can retard bone development. Vitamin C is involved in collagen synthesis. Deficiency of that can result in slender, fragile bones. Growth hormone, a very important hormone, stimulates cartilage cell division. Insufficiency of growth hormone can result in pituitary dwarfism, whereas an excess can result in giantism if the epiphyseal plates have not closed. But if that excess begins after the closure of the epiphyseal plates in an adult, the disease is called acromegalia, and the signs are different. Thyroid hormone will cause the replacement of cartilage with bone in the epiphyseal plate and increase osteoblast activity. Parathyroid hormone stimulates osteoclasts to break down bone. Sex hormones, including estrogens and testosterone, promote bone formation and stimulate the ossification of epiphyseal plates. And physical stresses, using your bones, is very important for strengthening your bones. Fractures. Fractures and breaks are the same thing. I'm going to tell you that on Monday, and it's one of my pet peeves. So we can have a simple or a compound fracture. In a simple fracture, the fracture is surrounded by uninjured skin or mucous membrane. It is a closed fracture. The skin is not broken. Whereas an open fracture or compound fracture is a fracture in which the bone is exposed through the opening of the skin or mucous membrane. It's pointing to the outside. It has broken that barrier. In addition to those two major types of fractures, the way the bone is broken can also lead to classification of different types of fractures. A green stick fracture is an incomplete fracture. One side of the bone is broken, the other side is not. You can really think of this as you've ever broken a branch off a tree, and when it's still green and you try to break it again, that's where the name green stick fracture came from. A fissured fracture is broken in the middle, the sides are still there. Comminuted fracture is really shattering of the bone into many pieces. Probably going to need some hardware to put that back together. Transverse fracture is across, oblique is at an angle, and a spiral fracture is caused when bone is twisted. That can be a sports injury when someone is changing directions quickly, but spiral fractures are also red flags for possible child abuse in young children. When a fracture is repairing, it starts with a hematoma, which is a clot. That's replaced by a cartilaginous callus. So we're going to put down a phagocytes removing debris. Fibrocartilage is going to invade, and that fibrocartilage is going to provide a model for the bony callus, in which the osteoblasts will come in. Hard callus will fill its place, and then remodeling will occur until you get back to the original shape, original structure before the fracture. And actually, after the fracture, it's healed completely. That bone right there tends to be a little stronger than the rest of the bones around it. It's not as likely to break it in exactly the same spot. Okay, we're going to call it there.